Hey everybody, it's Merm, and today's lesson is about cellular respiration. So I want you to open up the PowerPoint called fermentation, because fermentation is the chemical reaction that we're going to study and do a lab on. But to understand it, we really need to know about cellular respiration. So cellular respiration is how we use sugars. So especially sugars, we're going to use any different food. So if you're not eating sugars, your body will use proteins or fats or things like that. But how we use sugars to produce energy for cells. And usable energy in a cell is called ATP. So this is definitely going to be quite a biology lesson for you. So ATP is adenosine triphosphate. That is what cells use for energy. So when you think about it, you're eating food so that you can break down that food and get energy from it. Literally the reason why you eat. So the sugars in your food, think of sugar as like glucose. We're going to break down C6H12O6. All the bonds in that molecule, that organic molecule, can be used as they are broken. ATP can be built. So that's coupling of reactions. All right, so what's the difference between aerobic and anaerobic respiration? So aerobic uses oxygen, and it uses a cell organelle called a mitochondria, which I'm sure you remember from living in an environment. And anaerobic, no O2 used. So one of them is going to occur just in the cytoplasm, and one of them is going to occur in the cytoplasm and the mitochondria, and we'll get to a picture of that on the next slide. There is a huge difference in whether you're using oxygen or not. This one that uses oxygen is able to get 30 plus ATP per glucose. That's huge. And over here, you can only get 2 ATP per glucose. So let's think about that for a minute. If I'm a bacteria, think about my life, maybe moving a little bit, reproducing, but that's it. Not doing all the things that myself as a human am doing on a daily basis. I need to get that maximum amount of energy out of each sugar that I eat. So humans absolutely must be doing aerobic respiration. If you hold your breath, your body might need to do anaerobic respiration for an extremely short amount of time, but the majority of time we spend in aerobic respiration because we need this much ATP. All right, so let's talk about respiration, where it occurs. So we're going to draw a cell here. And so the main part inside of a cell is cytoplasm. And this is where step 1 occurs. So we're going to have 3 steps of aerobic respiration. Step 1 is for anaerobic and aerobic. And then here's going to be my mitochondria. So a mitochondria has multiple regions to it. So this is the outer membrane, and this section right here is called the outer compartment. Here, the membrane inside, this is called the cristae, and then the middle is called the matrix. So step 2 of aerobic respiration is going to happen here in the matrix, and step 3 actually involves both the cristae and the outer compartment. All right, so why is the mitochondria called the powerhouse? So you've always heard this, right? Mitochondria is the powerhouse of the cell. So why is it that? That is because this is where the greatest ATP production occurs. And this is what is needed for aerobic respiration. All right, so to get that maximum huge number, like 30 ATP per glucose, I need to have a mitochondria. The reactions have to happen there. So the steps of aerobic respiration. So remember this first step, glycolysis, this is just happening in the cytoplasm. And I've got to start with glucose because remember that's my molecule, that's my sugar that I'm breaking down. And I break it into what are called pyruvate, Pyruvate. Now, I like to show dots to represent carbons. So glucose is C6, so 6 carbons. So there's the 6 carbons in glucose. And if I break it into 2, each one of these has 3 carbons. So I haven't made any carbon dioxide yet. I have not done the major process of making breaking my glucose apart. Now, glucose is a very stable molecule, so it actually requires 2 ATP to start breaking it. It's not an easy to break molecule, but through the process of breaking it in half, it makes 4 ATP. So that we say is a net gain of 2 ATP. 2 energy molecules. So think about it like this. If you were working a little business and you had to put $2 in to get it started, but you're like, hey, I made $4. You didn't really make $4. You brought in $4, but it took you 2 to get started. So that's why we say your net gain is 2 ATP. And how do we get those 2 ATP? From breaking glucose in half. So by breaking glucose in half, you release a little bit of energy, and that's the 2 ATP. And that's what something like bacteria could survive on, just doing this step over and over and over again. It also does build up a molecule that is pretty awesome. I like to call it cash money. So it uses 2 NAD pluses. It reduces them. So remember, reduction is gaining electrons. So when you break glucose, a hydrogen and 2 electrons are going to go to this NAD, and it's going to change it into 2 NADHs. NADHs are coenzymes. They are molecules that your cells use as hydrogen ion and electron donors and acceptors. So they go back and forth being oxidized and reduced, oxidized and reduced, oxidized and reduced. So right here, this is reduction that is occurring because it is gaining electrons and gaining H pluses. And I'm going to use it later on. That's why I call it cash money. I want to build up these NADHs. These are a super important molecule. If I have a mitochondria, if I can do aerobic respiration, these are super important molecules. All right, Krebs cycle. So before I can do the Krebs cycle, I actually have to do this little step first. I have to convert my 2-pyruvate into 2-acetyl-CoA. This step happens in that matrix of the mitochondria, so in that middle part. And what it does, let's draw our carbons. What it does is it changes a 3-carbon molecule into a 2-carbon molecule. So where'd the other carbon go? Carbon dioxide. So since I start with 2 pyruvates, I'm going to break them into 2 acetyl-CoAs, and I'll have 2 carbon dioxides. But it also builds up that awesome NADH molecule that I was talking about. So 2 NAD pluses become 2 NADHs. Cash money, building up my cash money. All right, then the Krebs cycle. So once I do this step, I can change these into 4 carbon dioxide, because if you remember, my goal was to make every carbon from glucose into carbon dioxide. So that is really my product. I've now taken C6H12O6 and broken it into 2 and 4, that's 6 carbons, all separated from each other. So I've done the maximum breakdowns that I can. But really importantly is I'm going to make some more of that cash money. I'm going to change 6 NAD pluses into 6 NADHs. And now I have another kind of cash money called FAD. And I'm going to make 2 FADH2s. So put a big cash money next to this. So if we do the math, I now have 6, 7, 8 and then 2 more over here, 9, 10. I have 10 NADHs that I've built up, and I have 2 FADH2s that I've built up. So I have built up something that I will be able to use in the last step of respiration. Even though I have no more carbons left of my carbon dioxide, I've built up this cash money. So the last step is called oxidative phosphorylation. For this step, you need oxygen, and you obviously need a mitochondria. This is going to use that cristae. Actually, I like to draw a picture for this one. So this is my mitochondria, and here is that cristae, that membrane. Here is my NADH, and remember, I've got 10 of them, and I've got 2 FADH2s. Okay, in the membrane, there are proteins, and these proteins make up what's called an electron transport chain. And the electron transport chain ends in oxygen. Oxygen is there at the end of the chain. It's what we call the final electron acceptor. So the first thing I need to do is I need to oxidize NADH. I need it to lose its electrons. When it loses its electrons, first of all, it's going to go back to NAD+, okay? And the electrons, oh, and there'll be H's. When it does that, my electrons, let's do it in another color. My electrons can go boink, 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 boink through this electron transport chain. Now, they are high energy molecules. So those high energy molecules are able to move hydrogen ions, pump them out into this outer compartment. So I'm going to build up a whole bunch of H pluses out here. All right. And when my electrons make it through the whole chain, oh, I'm going to have FADH2 do the same thing. It gets oxidized. And as it's oxidized, I get the FAD back. So think about it. NAD plus, FAD back in the matrix. They're going to be able to go through the Krebs cycle again and again and again and make more and more and more. And keep doing this over and over and over again. My electrons at the end of the chain, if I take electrons plus O2 plus the H's that are out here, I make water. So I want you to do something for a minute. I want you to take your hand, put it over your mouth. When you breathe out, I want you to go like, breathe that air into your hand. You will feel some moisture. That's the water that you're making. So carbon dioxide, you are breathing out. You are breathing in oxygen for this process. You're breathing out the carbon dioxide that you made in the Krebs cycle and in that first preparatory conversion step of the Krebs cycle, and you're breathing out this water that you made by moving your electrons through this cash money. Now, the last step is super important because you might notice we haven't made any more ATP right now. So we are going to use a special channel protein. This channel is able to use these H's, and it actually has this little spinning part. So a protein is embedded in the membrane of the cristae. It takes these H pluses and it funnels them through. And they're moving from high to low concentration, so they're going with the gradient. And picture it like going through a water tube. They're going through, and this, or like a turbine, it's spinning and spinning and spinning. And as it spins, it puts together ADP and phosphate, and it makes ATP. So ATP is produced like crazy in this process because I build up a proton gradient out here in the outer compartment, and I use that to make ATP. And for every NADH that I oxidize, I can get about 3 ATP. So 10 times 3, that's 30 ATP I can get. FADH2, they donate their electrons a little later in the chain, so they can get about 2. These are a little bit overestimates, but that would give me 4. So I could get 34 ATP from just this oxidative phosphorylation step. Bacteria can't do it because they don't have mitochondria. We can do it because we do have mitochondria. That's how we maximize the amount of ATP that we get. I actually want you to go back to this slide. I just realized I didn't show you. We actually do get 2 ATP from this Krebs cycle step. It's not much, but at least we get a little, and that is again from a breaking reaction. You always couple the breaking of molecules with the building of molecules. So we do get 2 ATP from that. Okay, what if there's no ATP available? So if there's no ATP available, then we have to go through just 2 steps. So first of all, no Krebs cycle and no oxidative phosphorylation. So no major ATP production. Instead, you go through step 1, which is glycolysis. That'll give you 2 ATP. And then you go through a new step, which is fermentation, which is really something that in chemistry we want to study today, because fermentation is a really cool reaction that is used in a lot of food production. So like fermenting cheese and alcohol, all of those things involve fermentation. And fermentation doesn't give us anything great. What it does is regenerates NAD+. Because you'll remember, NAD+ was needed in glycolysis. If you use all of your NAD+ to make NADH, but then you don't have a mitochondria to use up that cash money, you're in trouble. You're going to run out of it. So the real goal is regenerating the NAD+. All right, and then alcohol versus lactic acid fermentation. So this depends on the enzyme available. If you have a, let's say a plant or a bacteria, they typically do alcoholic fermentation. And they make what is called ethanol. And if you have an animal, then an animal does lactic acid fermentation. And I want to show, okay, I want you to add a slide here. So go to your home button and click new slide. And you can just make it be blank, add a slide. Okay, so I just want to show you this chemical reaction because this is the reaction that we care a lot about. So if I take pyruvate, which is going to have 3 carbons, I can break it down into an alcohol that will have 2 carbons. So here's what ethanol looks like. 2 carbons. All right, and then I'll also release carbon dioxide. So that's where my 3rd carbon comes from, carbon there. Okay, so C3, so if I start off with 3 carbons, I can have 8 H's on there. And I'm going to have other molecules present because this used to originally be a sugar. So, there's going to be oxygens available as well. You could do it with C3H8, but I'm actually going to have some oxygens there as well. All right, and then last thing, conducting a fermentation lab. So we want to test this. So we want to see, we want to be able to compare. We're going to actually use different juices. And how much CO2 they produce. It's difficult to measure the alcohol. It's easier to measure the CO2 because it's a gas. So we actually have these little fermentation tubes. They look like this. It has a little bulb section to it. Sorry, that is a terrible drawing. Let me try that again. Let me erase that. All right, so it goes like this. Okay, that's better. So we're going to put our solution in here, and the solution will actually go up the tube. And then as it produces gas, we can measure the gas produced. All right, so we're going to be able to compare it by comparing how much gas they each make. And since we're starting with sugar, actually, go back to this slide. Let's erase this. Let's start with half of a sugar molecule. So a pyruvate would actually be C3H6O3. Okay, far more effective. And look, let's see, 3 C's, 1, 2, 3 C's, 6 H's, 1, 2, 3, 4, 5, 6, and then O3, 1, 2, 3. Perfect. So I want half of the glucose molecule that we start with there. All right, so let's erase this part. I want a pyruvate. So I'm labeling this is my pyruvate. And I'm going to make ethanol. So remember, eth for 2 carbons, enol because it's an alcohol. There's the alcohol functional group right there. And then this is carbon dioxide. All right, so as I run this experiment, I need to have it at a nice warm temperature. So we usually try and do this at around 42 degrees Celsius. So like that's a little bit warmer than your body temperature. Your body temperature, 98.6 Fahrenheit, is around 37.5 Celsius. So it's just a little bit warmer. It's little ideal conditions for it. So we will do that during the block, and we'll measure how much gas that we produced, and we'll compare our juices, and we'll compare how much sugar are in the juices too to see how much respiration we have occurring in the form of fermentation. All right, have a great day, everybody.